Okay, Assalamu alaikum. We want to talk about part 2, strategies of metabolism, oxidized and reduced coenzymes. Now, many metabolic reactions are oxidation reduction reactions. A steady supply of oxidizing and reducing agents must be available. Now, a few coenzymes cycle continuously between their oxidized and reduced form, and here are some examples. Now, the coenzyme nicotinamide adenine dinucleotide, this is an oxidizing agent. And this is a reducing agent. It's reduced form, oxidized form, reduced form. Nicotinamide adenine dinucleotide phosphate, NADP+, oxidized and reduced form. Flavin adenine dinucleotide, oxidized and reduced form. And the flavin mononucleotide, oxidized and reduced form. Oxidation can be loss of electrons, loss of hydrogen, or addition of oxygen. This is the meaning of oxidation, whereas reduction can be gain of electrons, gain of hydrogen, or loss of oxygen. Now, oxidation and reduction always occur together. Each increase in the number of carbon-oxygen bonds is an oxidation, and each decrease in the number of carbon-hydrogen bonds is a reduction. Now here, if you look at the structure of NAD+ and its phosphate, are coenzymes that enter and leave enzyme active sites in which they are required for redox reaction. And this is the site of reduction, you see here, and this is the reduced form. An oxidizing agent like NAD+, they remove hydrogen. They remove hydrogen from a substrate, and as a reducing agent, they provide hydrogen that adds to the substrate. Now, if we take this example, the oxidation of malate to oxaloacetate, you see, this alcohol is oxidized to keto group by malate dehydrogenase enzyme, and the oxidizing agent is NAD+. Now, the oxidation of malate to oxaloacetate requires the removal of two hydrogens, you see, to convert a secondary alcohol to a ketone. Now, the oxidizing agent, which will be reduced during the reaction, is NAD+, functioning as a coenzyme for the enzyme malate dehydrogenase. So it is both a coenzyme and an oxidizing agent. Now, a hydrogen atom is equivalent to a hydrogen ion, H+, plus an electron. So the other coenzyme called flavin adenine dinucleotide, another common oxidizing agent, is reduced by the formation of covalent bonds to two hydrogen atoms to give FADH2. So this is the site of reduction, you see here, by formation of covalent bonds to two hydrogens. This is the reduced form, and this is the oxidized form. So they form covalent bonds to two hydrogen atoms. Now, because reduced coenzymes have picked up electrons in their bonds to hydrogen that are passed along the subsequent reactions, they are often referred to as electron carriers. As coenzymes cycle through their oxidized and reduced forms, they also carry energy along from reaction to reaction. Ultimately, this energy is passed on to the bonds in ATP. Now, citric acid cycle. It is a cycle of 8 reactions where the acetyl groups in acetyl-CoA, the one in red, are readily removed in an energy-releasing hydrolysis reaction. As you can see here, delta G is minus 7.5. This is called thioester bond. It even gives more energy than the hydrolysis of ATP itself. Now, oxidation of two carbons to give CO2 and transfer of energy to reduce coenzymes occurs in the so-called the citric acid cycle, which is also known as the tricarboxylic acid cycle or Krebs cycle. Now, the citric acid cycle is a closed loop of reactions in which the product of the final step, oxaloacetate, is the reactant in the first step. So it is a closed loop, and this is how it looks like. You see here, it consists of 8 reactions. As you will see, we will consider them one by one, but this is a summary. In step 1, the acetyl group is transferred from acetyl-CoA to oxaloacetate to oxaloacetate to form citrate. In step 2, this citrate is isomerized to isocitrate. Then isocitrate loses CO2 from here to yield this alpha ketoglutarate, which then loses another CO2 from here. Notice that step 3 and 4 are called oxidative decarboxylation reactions. 2 CO2 were produced in 2 consecutive steps. In addition, NADH, 2 NADH are produced. Then here we have succinyl-CoA is the product. In step 5, this succinyl-CoA is set free to give succinate plus CoA in a reaction coupled with GTP formation. So notice here this thioester linkage, as I told you, is a high energy bond. When it's hydrolyzed, it produces enough energy to phosphorylate GDP to form GTP, and this is called substrate level phosphorylation. Then succinate, 2 hydrogens are removed from here to form the double bond, and this is an oxidation reduction reaction, as you can see, and we produce FADH2. In step 7, addition of water across this double bond to form malate, and then in the last step of the cycle, malate is oxidized to regenerate oxaloacetate again, and NADH is produced. And here again, these 8 reactions are catalyzed by 8 different enzymes, and here you can see in summary each enzyme and the reaction product. Citrate synthase produces citrate. Econitase produces isocitrate. You should know all these reactions and all these enzymes, and we will now talk about them in details. Now in steps 1 and 2, acetyl groups enter the cycle at step 1 by addition to 4-carbon oxaloacetate to give citrate. Citrate is a tertiary alcohol and cannot be oxidized. It is converted to isocitrate, a secondary alcohol that can be oxidized to a ketone. This isomerization catalyzed by an enzyme called econitase. Now both steps are oxidations that rely on NAD+ as the oxidizing agent. One CO2 leaves at step 3 and a second CO2 leaves at step 4. Now when you combine oxidation and decarboxylation, these 2 steps are called oxidative decarboxylation reactions. In both steps, electrons and energy are transferred. Succinyl-CoA carries 4 carbon atoms along to the next step. The 4-carbon oxaloacetate must be restored for step 1 of the next cycle. The exergonic conversion of succinyl-CoA to succinate is coupled, as I explained, with the phosphorylation of GTP to give GTP. Now GTP is similar in structure to ATP, and like ATP carries energy. In many cells, GTP is directly converted to ATP. Step 5 is the only step in the cycle that generates an energy-rich phosphate. In step 6, succinate from 5 is oxidized by removal of 2 hydrogen atoms to give fumarate. The enzyme for this succinate, dehydrogenase, is part of the inner mitochondrial membrane. Remember, step 6 is the only reaction in the cycle that occurred in the inner mitochondrial membrane. Why? Because this enzyme called succinate dehydrogenase is found there, not in the mitochondrial matrix. The reaction requires Fad, which is covalently bound to its enzyme. Succinate dehydrogenase and Fad pass electrons directly into electron transport. Now step 6 neither uses nor releases energy. In step 7 and 8, water is added across the double bond, as I showed you. Then oxidation of secondary alcohol to give oxaloacetate. And here you regenerate the starting material, and this will keep the cycle on, because the regenerated oxaloacetate will pick up a second molecule of acetyl-CoA, and the cycle continues. Now the rate of the cycle is controlled by the body's need for ATP and coenzymes. When energy is being used at a high rate, ADP accumulates and activates isocitrate dehydrogenase, the enzyme for step 3. Now when the body's supply of energy is abundant NADH is present in excess and acts as an inhibitor of isostrate dehydrogenase. Now the cycle is activated when energy is needed and inhibited when energy is in good supply. So this is the case. So the enzyme, in summary, isostrate dehydrogenase, as you can see, is inhibited by excess of NADH and activated by ADP. Even ADP and NADH are allosteric effectors of the enzyme called isostrate dehydrogenase. Now, at the conclusion of the cycle, the reduced coenzymes are ready to donate their energy to making ATP. The energy is released in a series of oxidation reduction reactions that move electrons from one carrier to the next. Each reaction in the series is exergonic. Now, the sequence of reactions is known as the electron transport chain, also the respiratory chain. The enzymes and coenzymes of the chain are embedded where? In the inner membrane of the mitochondria. Here you can see we have 1, 2, 3, we have 4 complexes. 1, 2, 3, 4. These are 4 membrane-bound complexes that are components of the electron transport chain. In addition, we have this is not part of them, but this is different, called ATP synthase. This is an enzyme that link oxidation and phosphorylation together. So here, for example, when electrons are transferred, protons are pumped from the matrix to the inner mitochondrial membrane. As you can see, this is the matrix. Protons are pumped in complex 1, not in complex 2, in complex 3, and in complex 4. As a result of electron transport, protons are pumped out of the matrix to the inner membrane space. Now, when these protons come back through the ATP synthase, this is the complex, enough energy is produced to phosphorylate ADP to form ATP. This is the idea. Now, notice here, complex 4 is the one that reduces oxygen to water. Then, the final electron acceptor from complex 1 to 2 to 3 to 4 is molecular oxygen, where it is reduced to water. So water formed here, and in the citric acid cycle we saw CO2 produced. So this is when we say, for example, that glucose is oxidized all the way to CO2 in the Krebs cycle and to water here in the electron transport chain. So the enzymes and coenzymes of the chain are embedded, you see, they are embedded in the inner mitochondrial membrane. Complex 1, 2, 3, 4. In addition to this called ATP synthase, which links oxidation and phosphorylation. Now, as electrons move down the electron transport pathway, the energy is used to move hydrogen ions from the matrix to the intermembrane space. Now, because the inner membrane is otherwise impermeable to the H+ ion, the result is a higher, so proton gradient is formed. Now, moving ions from the region of lower concentration to one of higher concentration requires energy to make it happen. The energy is recaptured for use in ATP synthesis. Electron transport proceeds in 4 enzyme complexes, as I showed you, in fixed position, complex 1, 2, 3, 4, within the inner membrane of mitochondria, and 2 electron carriers that move from one complex to another. The 4 fixed complexes are very large assembled bags of polypeptides and electron acceptors. Electron acceptors are generally cytochromes or quinones. Here we have oxidized coenzyme Q, the reduced coenzyme Q, and the heme group. Again, these are the 4 complexes, complex 1, 2, 3, and 4. Now each of complexes from 1 to 4 contain several electron carriers. Now hydrogen ions and electrons move through the pathway in the direction of the arrow. Here, this is the direction of the arrow. Then hydrogen ions and electrons, they move in the direction of the arrow. Each complex is at a lower energy level than the preceding one, and this allows the spontaneous flow of electrons, okay, from high to low to low to low. Complex 1 contain an iron sulfur protein. There is, you have coenzyme Q. This is called a mobile electron carrier. Coenzyme Q carries the electrons in addition to cytochrome C, as you can see. So we have, these are electron carriers. One is called CoQ and the other is called cytochrome C. Now plant cells contain mitochondria and chloroplasts. Again, plant cells contain mitochondria and chloroplasts. Chloroplasts carry out photosynthesis, a series of reactions that also involve an electron transport chain. Now, hydrogen ions can return to the matrix by passing through the channel that is part of the so-called ATP synthase enzyme complex, and in doing so, they release the potential energy gained as they were moved against concentration gradient. This energy release drives the phosphorylation of ADP. Now, recent research suggests that each NADH molecule is about 2.5 molecules of ATP, and that each FADH2 molecule yields approximately 1.5 molecules of ATP. And this is the one I want you to know. This is called the P to O ratio. P, number of organic phosphate, and O for number of oxygen atoms that are reduced to water. So NADH yields 2.5 ATP, and each one FADH2 yields 1.5 ATP. I want you to know this. Finally, there are the so-called blockers and uncouplers of oxidative phosphorylation. Now, blockers like cyanide and barbiturates are among a group of substances that block respiration, or the so-called oxidative phosphorylation at the cytochromes in the electron transport chain, with different blockers acting on different cytochromes. Also, you can add here CO poisoning by CO, carbon monoxide, and this is very dangerous in winter, where people can suffocate or get poisoned by this poisonous gas, which evolved from incomplete combustion reaction. In addition, these are two blockers. We have also the so-called uncouplers. Uncouplers like 2,4-dinitrophenol and valinomycin. Antibiotic valinomycin allow electron transport to occur but prevent the conversion of ADP to ATP by ATP synthase. So these are they uncouple oxidation from phosphorylation. So when ATP production is severed from energy use, it is said that ATP production is uncoupled from the energy of the proton gradient. So however, the effect of uncouplers is temporary. If uncoupler is removed, then ATP production is resumed. This is the end of part 2. Salam.